Okay, so today we are going to cover our last topic of acarology, which is mites. Earlier we have talked about the ticks in details, and the ticks briefly we know that they were having an exoskeleton on them, and they were having a scutum or conscutum on their bodies. They were quite general characteristics for the order Acarina, which includes both of them. So the mites, these are called as mini hairy ticks. That's why we are just referring it back to the ticks once again. That they are resembling with the ticks as far as the morphology is concerned. They have the similar kind of mouth parts as we talked about earlier. That they are labrum, labium, mandibles, and hypopharynx. They are all replaced with the structures that we mentioned about the palps or the pedipalps, and then a pair of chelicerae and medially placed or ventrally placed hypostome. Then the mite larvae are given due to their hairy nature. They have been given a specific name that is called as the chiggers. And they do not feed on blood basically. They secrete digestive fluids into the host skin, and they are feeding on the dead or decaying organic matter of the body. And some of those, I will talk to you later, that some of those are referred to as the burrowing mites because they make tunnels into the skins. Okay. Briefly about the difference between ticks and mites. Although they are belonging to the same class, which is class Arachnida, there are some differences. Like the hypostome in ticks is toothed and it is exposed, which means it is visible. Why is it toothed? We make a presumption that because they have to penetrate into the skin to take up the blood meal, और इस वजह से उनके जो hypostome हैं, they are provided with teeth-like structures so that they can facilitate in the penetration process. And as we mentioned that the mites are not feeding on the blood, they are just releasing the digestive juices to be secreted at the point where that is causing proteolysis, and all the digested stuff is going to be taken up by the mites later on. Ticks are large, they are easily macroscopic, they are visible with the naked eye, and we mentioned about their sizes. The original size is increased up to 200 times when they are repleting, when they are feeding on the blood. But this is not the case in case of mites because they are small and usually they are microscopic, because we have to go for skin scraping examination. You might have studied in your practical examination, practical part of the syllabus, where you have to scratch the skin with the help of a scalpel, making it an angle of 90 degrees, so that you can pull out the mites which are hiding somewhere in the deeper layers of the skin too. And you are scratching the skin up to the point when the blood is oozing out from that location, and that is the point when you are presuming that you have probably reached up to the level where the mites can be examined and can be pulled out. And you are placing that on the slide and then examining under the microscope for visibility of the adult mites as well as their developing stages. The Haller's organ are present on the first tarsi. We have talked about that, which was having a sensory function, which was being utilized by the ticks in order to sense the opposite gender, in order to sense the chemical stimuli in their vicinity. However, mites they are not lacking this Haller's organ. The peritreme, you know, there is a peritreme in the stigmal plates, the outskirts of the stigmal plates is called as the peritreme. It is absent in case of ticks, but here the peritreme is present in mesostigmata. Here I am talking about the adult ones, and in the adult ones, the body is having a peritreme which is circling on both sides of the head and reaching up to the middle of the body. दोनों साइड्स पर हेड के एक पेरिट्रीम एक मार्जिन है जो कि रन करता है, and it is reaching up to the middle of the body in adults I'm talking about. That is called as peritreme. I'll show you in the picture. Okay. Okay, here. So we already talked about this differentiation, this division actually, the palps and the chelicerae, and this is the gnathosoma and the prophotosoma, and then the idiosoma. These are and opistosoma. There are different names in different vocabularies in different textbooks. But here, a few things which are visible are the hairy nature of the body, and then the ends of the legs. Here we can see the three terminal part of the legs, which are having some kind of suckers in mites. Now in mites, the shape of the sucker itself can be one of the differential characteristic based on which you can differentiate various species of the mites. But in ticks, this was ending up in the claw, and that claw was having a web-like appearance, which was called as pulvilline. They are having parasitic and free-living forms, because all of the stages are not parasitic, and they are small size, most being less than 0.3 millimeter in size. Some of those are blood-sucking species may attain 1 centimeter. They are very small and they reach very deep into the skin to reach up to the bloodstream. But most of the times they are not relying on the blood. With few exceptions, they remain in prolonged contact with the host, causing mange. Now, in ticks, we talked about that they are attaching with their host for a maximum of 15 days period. During that period, we divided that into various phases. There was an attachment phase, there was a slow feeding phase, and then there was a fast feeding phase. And eventually it has to drop off. And this was the case in soft ticks when they were coming and then were dropping off. Here, because they are microscopic, they are not going off their host. They are spending, in some cases, entire life cycle on the host, on the skin, or maybe inside the tunnels which they have been making while they were on the host. So, prolonged contact is there, and that prolonged contact make a very challenging situation for the practitioners that how to treat them. Because whatever drug you are applying, that may not be able to reach up to the deeper tunnels, and the cure rate is quite questionable in some cases. So the situation or the condition which is being caused by this prolonged contact, which is persistent pruritus and inflammation of the skin, that is rendered as the mange in animals. So it is also called as scabies in human beings. You might be familiar with that. So most species spend entire life cycle on one host, and it makes sense that if they are spending entire of their life cycle on the host, the transmission is possible through the physical contact. Burrowing mites and non-burrowing mites, these are the two terms. We'll talk about that later, and there is another slide where we have mentioned it. So end of the legs, the pretarsi consist of hook, a stalk, and a terminal sucker. That's what we mentioned in the picture. That the terminal part of the leg, which was in ticks ending up in the claw, here they are consisting of a hook-like structure, and that hook is connected to a stalk, a long filament, and then a terminal sucker. Actually, they have this thing is a little bit detail of this particular part, that it is ending up in a hook, and then there will be a short pedicel, and then or the stalk, and then it ending up in the sucker. All right. There are 3 octopode nymphal stages. In the life cycle, they have 3 nymphal stages, which are also termed as protonymph, deuteronymph, and tritonymph. And all of these 3 nymphal stages are having, octopode means 4 legs, 4 pair of legs. Females burrow inside the skin and lay eggs, which are 3 to 5 eggs daily, and a total of 40 to 50 eggs are being laid in their life cycle. They are very susceptible to dryness and cannot live off the host. So if they will be trying to even going off the host, due to dryness or due to adverse environmental conditions, they will die off. So they need to have moist and humid environmental conditions for their survivability on the body. So a brief about the life cycle, composed of the egg, which is hatching to 6-legged larva, which is now here called as the chigger. Then it's converting into 4-legged nymph, and then now the nymph is having 3 nymphal stages, and then it is converting into male or female adult. As far as the mites, the medical importance of the mites is concerned, it is causing scrub typhus, which is dermatitis with severe itching, and this itching can end up with pustules or papules like this on the extremities mostly. It causes lower production in case of animals, because the animals will keep themselves busy in scratching their bodies. And then the intermediate host of various cestors are also among the significant points that they are causing transmission of a wide range of cestors to their definitive hosts. Now here's the deviant. 2 major deviants, burrowing and non-burrowing. Burrowing mites are those, as I mentioned, that they are making tunnels in the skin. They are, with the help of their proteolytic enzymes, they are making tunnels so that deep inside the skin they can reside, and not only reside, they can eat up the necrotic tissues as well as lay eggs at that point. And that is, increasing the chances of their survivability on the host. And there are 4 important species, which is Sarcoptes, Notohedrides, Nematocoptes, and Demodex. These 4 genera are within the category of burrowing mites, and then Coreoptes, Sauroptes, Saurogates, and Dermanices. These are the 4 mite species, although there are many others, but these are very important. These are under the category of non-burrowing mites. So about the non-burrowing mites, they are residing on the fur or the hairs, and whenever they have to take the meal, they are diving deep into the fur to reach the skin, causing some feeding activity, feeding up the stuff, and then going back again on the fur or the hairs. So they are not residing deep into the tunnels. So a little bit about Sarcoptes, because this is one of the most important members in the burrowing mites. It parasitizes wide range of mammals, important for humans as well as animals, and in man it is causing scabies. And Sarcoptes scabii is the species which is causing the scabies in human beings. It is a round mite up to 0.4 millimeter in diameter only. So they are very small, as I mentioned, microscopic. With transverse ridges and triangular scales. So these things will be visible when you will see the picture, that what kind of ridges and scales are there. Look at that. So these are the ridges which we mentioned over here. These lines are representative of the ridges, and transverse means across the length, and if we say longitudinal, they are along the length. So transverse ridges and some scales. These triangular things are indicating some scales. So these are the terms used to explain their morphological features. Now here, differences tarsi of first, second, and fourth pair of legs in males, and first and second pairs in females are having bell-shaped suckers. So once again, so if we closely look over there, the ending part of the sucker looks like a bell, and which is in the statistics you might be understanding the normal distribution. So that is actually called as the bell-shaped curve, because it looks like the ringing bell. So at the end, these are having the bell-shaped suckers, which are narrower at the base, and as soon as they are proceeding towards the terminal part, they are wider. So that is the morphology of the sucker in Sarcoptes. And it is also indicating sexual dimorphism, that in males these suckers are only present in first, second as well as fourth, but in females it is only present in first and second. These suckers are called as carandules. These suckers have been given specific name for that. So the suckers which are devoid of the tarsi which are devoid of the suckers, they are bearing additional structure which is called as the bristle. ऐसे tarsi जिनके ऊपर suckers नहीं होंगे, so they would be bearing bristles. So pedicels, what are pedicels? Filamentous stem connected to tarsi bearing sucker. We already talked about that because it is the morphology of the sucker at the end, which is having a hook terminal part. Along with that is the pedicel or the stalk that you mentioned, and on the stalk is connected with the cell. There is no ad-anal suckers which are found. Why is it so? Because in some of the mites there are some suckers which are adjacent to the anus. We mentioned about that in ticks, that there are some plates which are present adjacent to the anus. These were called as ad-anal plates. So third and fourth pairs of legs do not project beyond the margin of the body. What does it mean? That the pair of legs are though very stunted in length. Stunted means that they are not elongated, as the ticks are having very long legs which are projecting wide apart from their main body. But in mites, they are so stunted that they are not even going out of the margin of the body. जो body का margin है circular, उससे बाहर भी नहीं जा रही legs, because they are not swift walkers. They do not need them probably, because they are just wherever they want to go. They are just so creepy that they just want to enter into the tunnels. That's it. But in ticks, they have to get off, then again reach to the host. So probably there is more involvement of the legs, and that's why their use and their morphology is different from there. The areas on the body where scabies mites are found are the hands, wrists, elbows, feet, ankles, penis, scrotum, buttocks, armpits, and so on. This is based on some investigation or study which was conducted, where the proportional prevalence of mice was observed in various parts of the body. So it was found that mostly the areas, almost most of the areas of the body are covered here, but the probability is much higher on the wrists and elbows as compared to the others. In animals, predilection site of mange are ear, muzzle, face, and elbows. So predilection site means, once again, the sites which are being liked by the parasite for their growth and propagation. So wherever the site is, over there it is causing erythema. What is it? The redness. It is leading to papule formation, which is it's kind of a vesicle, inside which could be a fluid, and that can end up with the pus. Scale and crust formation. Those papules will be bursting, releasing out the fluid inside that, and that end up with the scale formation and crust formation over there. And all the affected areas will be suffering from alopecia. So there will be, due to this activity, there will be intense pruritus, which is inflammation of the skin, the scratching of the skin, and this scratching can lead to self-inflicted trauma. So this pruritus is exacerbated, it is exaggerated due to hypersensitive reaction to mite allergens. Because one thing is physical presence of the mite itself, which is causing mechanical damage. It is causing the host to scratch the locality. Second thing is release of the antigens at that site, and the antigens themselves are eliciting strong immune reaction into the body, and the host is also trying to get rid of those allergens somehow. Ultimately, weak and emaciated animals would be resulted, and there would be, due to all this secretory process going on at that point, there would be sour odor, which is a notable feature of the mite infestation, which is a fetid odor coming out of those crusts or the scales. So in dairy animals, the hides, they would be downgraded. The hides would be of lower quality because of the involvement of the skin, making of the tunnels. there will be lower production. These scabies mites are extremely contagious. It is also observable in human beings that if a single person in the household is suffering from scabies, probably there's a higher chance that most of the households would get a chance to get infected. So if one person is getting treatment, mostly all others are getting treatment as a preventive pathogen spread. It is often assumed that the family or living group should also be treated. At the very least, infested persons should sleep separate from the other persons. Definitely, it is, as we mentioned, it is a contagious thing. So contagious means it is spread from contact, or any possible contact should be avoided. So here is the image where we can see on the anterior side, this is the head region, and these are the legs. Look at the dorsal side. Can you see the terminal pair of legs? No. We can see only two anterior pairs of legs. The terminal pairs will only be visible on the ventral side of the body. These are dorsal sides of the body. This is the ventral side. Here you can see. These are the two very limited sized, stubby kind of appearance of the posterior part of the legs. Okay, so canine scabies is also caused by Sarcoptes scabii. Notoherpes cati. This might infest cats. Somewhat similar in behavior and pathogenesis to Sarcoptes. It is behaving similarly as far as the ecology is concerned, but it has more restricted host range. So it is primarily causing infestation in cats. But occasionally it can be seen in the temporary parasite in other species, including human beings too. It is distributed worldwide, and it is also morphologically also resembles with Sarcoptes, having a circular outline and short legs, as we have seen over there. But how is it distinguished? Distinguished by its concentric thumbprint striations. What was the kind of striations earlier? They were transverse striations. And now, look at this. Concentric, making circular fashions. And then, it is also devoid of, and one thing else, which is absence of the spines. Previously, in the Sarcoptes, we talked about that the whole body was covered with spines and bristles. Yeah, over here you can see the bell-shaped suckers are also prominent. And in a similar fashion, from the dorsal side, you cannot see the terminal two pair of legs. The life cycle is also similar, except that the females in the dermis do not occur singly. They are found in aggregations in cats. That is the only difference, and these aggregations are called as the mite nests. What is the pathogenesis? It is highly contagious, as we have talked about in Sarcoptes, but though it occurs in local limited outbreaks, it is not a frequent cause of skin disease in cats. How does the infection appears? Infection is usually dry, encrusted. Encrusted means it is also showing some kind of dried necrotic part on that vesicle or on that pustule, whatever is being formed by these mites. These are scaly lesions. Again, the same kind of explanation for the lesions which are being made by the mites. On the edges of the ears. Now the ears are more about the predilection side, maybe on the face, maybe on the skin, which is also showing thickening. So ultimately, what is the pathogenesis? The thickened skin, which is having a scaly surface. The associated pruritus is often intense. Now, one sense could be because of the nests, that there is not only a single mite at that location, there is an aggregation of mites in those nests which can cause pruritus, and the intensity would be severe as compared to the Sarcoptes. And there may be a severe excoriation of the head and neck from the scratching. Excoriation means sloughing off of the topical layers of the skin because of severe scratching or pruritus. In typical cases, the lesions appear first on the medial edge of ear pinna and then spread rapidly over the ear, face, eyelids and neck. So here we can see that the pathogenesis starts from the ear pinna, the central point, and then they are spreading towards the other parts of the head region. And then it can also be spread to the other parts when the cat is having the habit of grooming itself. So the ears are in contact with the other parts of the body, maybe the tail and the other parts, maybe the inguinal areas during licking and scratching. So they can be transferred very easily. How these to be diagnosed? Definitely the host itself. First impression, if the cat is coming to you, the mites that come into your mind must be the notohedrous species. But you should not be, confirmatory about that. You should go for skin scraping and examine for the morphological features as you have studied. The intense pruritus, the complaint is also one indicator. And then rapid spread to involve all kittens in a litter because of their magnitude, because of their more volume of mites on the skin. There are chances that more and more kittens in a litter could be getting infection. And confirmation is always by skin scrapings, which is more readily achieved than the sarcoptic mange, since the single nest yields many mites. So यहाँ पर क्या होगा कि if you are doing for skin scraping examination, the probability of getting more number of mites would be there because of, again, the same thing which is making the nests. How do we treat it? So here, skin crust should be softened with liquid paraffin so that you can reach up to the skin which is hidden below that crust. Because otherwise, if you are just applying everything on that crust, that is not allowing the chemical or the drug to reach to the skin where it is actually required. So softening of the crust is required with liquid paraffin, and along with that, after that you can apply some suitable acaricide. You can also apply 1% solution of selenium sulfide, which is recommended in cats. Why? Because organochlorines can prove toxic to cats. They are somehow, that's why contraindicated in cats. Treatment should be given at weekly intervals so that the developing stages of the mites should also be covered. Otherwise, what will happen if you are not repeating the drug? The adults may be died off, but again, the mites which are now recently coming out of the eggs, they would be spreading the infection. So repetition of the drug is very necessary here. Although not licensed, because the ivermectins are also contraindicated in case of cats, but in the controlled environment, ivermectins also showing efficacy against these mites. Nemidocoptes, it is the only burrowing mites of the domestic birds. And it is having two species, mutans and gallinae, and these are involving two different parts of the bird. Like the part which is infested by Nemidocoptes mutans is the leg region, which is the leg mite beneath the leg scales. You know, the chicken, they have scales on their legs. So these mites, they are hiding themselves just under those scales and then spreading them inside. So they are causing lameness, distortion of the feet, and as well as the claws. Then the gallinae, they are deplumbing itch mite because, as the word indicates, they are hiding themselves in the feathers. And due to severe scratching, the birds may be losing their feathers. They are trying to scratch those feathers due to this deplumbing itch mite, and there will be alopecia of various regions which are infected. Again, it is having a circular body, short legs, and avian hosts. These three things are sufficient to diagnose Nemidocoptes. They are having short and stumpy pedicels. Again, the pedicels just before the sucker. Three parts, which were what was those? The hooks, pedicel, and the suckers. All legs of male possess suckers. Now here's the difference. While none of the females do so. So again, it is Sexual dimorphism, that based on the presence or absence of the suckers, you can easily differentiate which one is the male mite and which one is the female mite. So, how to control that? Dipping of the legs in the acaricide solution can help, and cleaning of the poultry housing and proper spraying with suitable acaricide is also effective strategy. This is how it looks like. Look at the stubby appearance of the legs. Can you see that where the legs are? These are very stubby over here. You cannot even see them. So in Galini, again, little bit higher, larger in size, but still very stubby. Look at that. This is the declining itch mite on the feathers. And this is the situation in case of legs, scaly legs. Now the legs, this is a common thing in those birds which are having a congested environment, in moist environments where the hygienic practices are not very good. So the legs will be easily getting infection from the environment and causing this kind of crusty appearance. Demodex in dogs. All mammals in hair follicles and sebaceous glands may get this Demodex species, and this is very kind of unique in its morphology. This is elongated and having a tapering body, just like the maggots or the bots that you have studied in class Insecta. They have short, stumpy legs. So in canines, after the name of the Demodex, the situation is called as canine demodicosis, which is due to penetration into the deeper skin tissues where the surface acting acaricides cannot access and end up with a severe situation, and we need to give the proper name, which is called as the canine demodicosis. A notable feature of all types of Demodex mange is absence of pruritus. It is quite surprising because of their elongated nature, because of their short legs and their penetration deep into the layers, so that the topical layers are not being affected somehow. And they are also, in addition to that, somehow controlling the immune system of the host by releasing some immunosuppressant agents. And those immunosuppressant agents are not allowing the host to act against them and not even feel something is wrong going on at that point. And that's why probably the itching or pruritus is very mild in this situation. So there are two forms. One is squamous and the second one is pustular or polysular. So it is based upon the level, what kind of losses and what kind of pathology is being done by them. In the squamous demodicosis, this is less serious. It is a dry reaction in which there is little erythema, but the alopecia is widespread. And there is desquamation and thickening of the skin. Desquamation means sloughing off the skin because of its dryness. And thickening of the skin because the immune system of the host is reaching over there, and it end up with some kind of a fight between the antigens and as well as the host cells, and that end up with the thickening of the skin because it includes the healthier skin as well as the necrotic skin which is on the top of that, and ultimately the skin becomes thickened. In some cases of this type, only the face and paws are involved. In the second type, which is the pustular one, demodicosis is severe, which is usually following a bacterial invasion. Now it is actually a combo or comorbidity of the mite infestation with bacterial infection, which is mostly caused by the staph. Here there are thickened skins, as well as there is pustule formation, which is actually the formation of those vesicles which are filled with pus, and from where serum, pus and blood that starts oozing out. And ultimately this situation leads to a kind of red mange, and as well as these kind of activity of oozing of these three things can end up with an offensive mousy odor at that point. So that is also typical of this demodicosis. So due to this situation and its deep penetration, prolonged treatment is necessary, and survivors may be severely disfigured because of this thickening stuff and a lot of activity going on due to bacterial infection. So that early euthanasia and sometimes is referred, because the disfiguration is up to that extent sometimes that the owners, they request you to please go for euthanasia. Euthanasia, do you understand what is that? Yes, peaceful, peaceful death due to some chemicals. This is a picture how it looks like. It is not oval, it is elongated, having very short, stumpy legs, and the legs are very hardly visible from the dorsal aspect, but they are visible on the ventral side. There are many species, which indicates that it includes humans, canids, bovines, goats, pigeons, rabbits, sheep, pigs, and as well as equines. But the point is, the names have been given after the name of the species, but they are not host specific. It never indicates that only this canis is found only in dogs. It is switchable to other hosts too. These are various stages of the mites. We can have the you can see the deutonyms here. We can have the protonym, deutonym, and here is the egg and the larva and nymph and adult. How to control? Individual control, proper removal. There are certain reservoirs. As I mentioned, that there is a wide range of hosts. So the reservoir is that host which is maintaining the infection in the environment. And whenever you are eliminating the infection from your target hosts, the host which you are focused on, which means the livestock, cattle, buffalo, sheep, goat. So there is a chance of spillover of the infection from that host. So those kind of hosts are called as reservoir host. Any good example for reservoir host? Reservoir host could be the crows, the sparrows, could be rodents, lizards, bats, sometimes the feral dogs and cats, which are not under your focus. These are reservoirs, which are successfully maintaining the infection, and whenever you are trying to eliminate the infection from your focused group, the chances are spreading of the infection from that particular reservoir. So that's why rodent and wildlife control is very necessary, and then chemical control by registered acaricides is also there. So very quickly about the non-burrowing mites, Sauroptes, Coreoptes, Otodactys, Chelicia, and Dermatitis. And the point is that you should be able to differentiate, I think, these Sauroptes and Coreoptes from the Sarcoptes one, because we know that Sarcoptes is burrowing, and these two members, they are non-burrowing. So let's see what these are. Sauroptes mice in sheep, as well as cattle and equines. There are different names, like Ovis in sheep and cattle. Again, host specificity is not there, but the names have been given after the name of the hosts. Equine in canines, and cuniculi in equines as well as rabbits. They are all oval in shape, and they are a little larger. Earlier we have talked about 0.4 millimeters. Here they are a little larger, about 0.75 millimeters, and legs are projecting beyond the body margin. So this is again a point. In Sarcoptes, the posterior two legs were not projecting beyond the body margin. Here we are at least able to visualize them on the dorsal side. We'll see in the next slide. They have pointed mouth parts, rounded abdominal tubercles of nails, and there are 3 jointed pedicels bearing funnel-shaped suckers. Now funnel-shaped is a little elongated. Funnel is different from a bell. Funnel-shaped are just like the teeth, just don't get a funnel, which is having a straight line and then suddenly it is opening up. While the bell is like making this kind of angle, which is not very rigid, it is smooth. We'll see in the next slide. Now, the presence of suckers is also different. Males are having suckers on the pedicels of first, second, and third. In the Sarcoptes, it was 4. While in females, in Sarcoptes, it was first and second only. Here it is first, second, and fourth. So the variability exists here. So dorsal surface is devoid of scales and spines. We have seen them in Sarcoptes, right? and it is having fine striations. Males are having a pair of copulatory suckers on the ventral speck and terminal tubercles bearing several hairs. So males, in addition to these suckers on the tarsi, are having another pair of suckers just close to the adigus. You know what is adigus? That is a name of penis in case of arthropods. The life cycle is typical of non-burrowing mites, which is not very different, that the non-burrowing mites are spending entire of their life cycle on the host, but the adults are sitting on the fur or the hair, and they are just reaching to the skin whenever they need something to eat. Pubescent females, posterior copulatory tubercles are also there. And in males, which is in females, these kind of tubercles are actually helping the females for copulation with males, which is lasting for 1 day, and after 2 days periods, ovigerous females, which is ready to lay eggs now, it is now lacking those tubercles. Now, this presence of tubercles and absence of tubercles is an indicative of that whether the mites are, the female mites are in the pubescent stage or they have already laid eggs and mated. This is also one sign of that. So after that is the egg laying process. The females, they are laying around 90 eggs during the lifetime of 4 to 6 weeks. And that's why over here you can just have a clue over there that after 4 to 6 period of time you need to have a repetition of the treatment. Because if you are controlling the adults, after this much period of time, the young ones would be ready to, and they would be needing a control measures. Development from egg, larva, nymph and adults takes around 10 days. Greater pathogenicity of this mite is attributable to the fact that unlike burrowing mites, it has piercing and chilling kind of mouth parts which causes severe damage. So the burrowing mites, they are just burrowing with the help of their proteolytic enzymes. They are not having very sharp mouth parts. But here they are not making tunnels, they are not burrowing inside the skin. Instead, they are having the projected mouth parts with the help of which they can cause severe damage, because now they have to just rely on the topical part of the skin. And every time they are reaching on the topical part of the skin, they need some tools to slough off the epithelium so that they can eat the dyed tissue from there. So that's why their mouth parts are a little more advanced. The sheep scab, the same thing. It is seasonal in pubescent and reservoir sites, और ये sites दी हुई हैं जहाँ पर ये more pronounced होती हैं, और ये seasons दिए हुए हैं during spring, summer and early autumn. So during the winter season, it can spread to the rest of the body, because winter is more likely, the parasites in general, they want to hide themselves. They do not want the winter season. Though non-burrowing, but active in keratin layers causing direct damage to the skin. And what actually is happening? There is zone of inflammation where the mite is present, that ends up with the vesicle formation. And from that vesicle, in the similar fashion as we talked about earlier, there is serous exudate coming out of that. As the lesion spreads, जैसे ये lesion फैलेगा, the center becomes dry because the host immune system is also playing its role. The center will become dry and yellowish crusty, while the borders where the mites are multiplying would be moist. So it would be something like that the center would be drier and all the portions away from the center would be moist because the mites would be in the spreading fashion. Initially there will be patch of lighter wool in sheep, and as damages enlarges, it can also end up with the self-inflicted trauma, and ultimately there would be ragged and stained wool. And I hope you understand what is the meaning. Rag means there is devoid of the wool, which is ragged, which is not having the fine hair follicles or the wool fibers, and it is also stained, which means that it is having staining of this exudate, serous exudate, and then shedding from large areas. What does it cause? Restlessness, off-feeding, suppression of the weight gain, and then scumbling of the animals. The animals will try to hide themselves, will hide to group among one another, making a group at one corner, and they will not be active. debilitation or the weakness, and sometimes cannot compete with the winter season, and they, at self now they would be now so-called immunocompromised, and that immunocompromisation will cause death of the animals due to winter, coupled with the mite infestation. In the quiescent phases, which are apparently healthy sheep, quiescent phase means that you cannot see the clinical signs and symptoms. It means it is subclinical mite infestation. In those cases, outbreaks occurred during the cold season. And then what will be done? Like dipping of the infected animals is the best choice. And do you know what is that dipping? Passing of the animals through a certain height of solution, which is a solution mixture of calicide in water as per the recommended concentrations. Coreoptes and Otodactylus, both of them, they feed superficially. We already talked about that. They do not have piercing mouth parts. They can only chew. They can shed the scales, and they can use the debris for eating purposes. They can infest a wide range of hosts. The species name can be given after the name of the host, like the Coreoptes bovis. Here the pedicels are short and unjointed, and they are ending up with the cup-shaped suckers. Now another third category, which is a little wider shape, not even like a funnel in the Coreoptes, not even like the bell in the Sarcoptes. They are having cup-shaped suckers. Now look at the arrangement. Pedicels of the legs of males, first, second, fourth pair, and all legs in males, and first, second, fourth in case of females. Again, the difference lies. Now Coreoptes has to be differentiated well from Sarcoptes, right? And as well as Sauroptes. Mild infestation as compared to Sauroptes, it is of milder nature. In horses, it is causing mostly the mange in the foot region. That's why it's called as the foot mange. Or it's called as the itchy leg. It is mostly affecting the neck, tail, udder and legs of the cattle, and rubbing and scratching, understandable. And in sheep, it can cause, in severe cases, can be pustular. And whenever the word pustular is shown here, it means that there is a pus formation. So dermatitis is inflammation of the skin. Now adding pustular means dermatitis coupled with the pus formation means pus vesicles. So these things are ending up with the wrinkling of the skin in sheep and thickening of the hair. And always dipping with suitable acaricide is the solution. Now this is a quick comparison. Sarcoptes, a long pedicel, right, and a sucker. Here it is bell shape. Then Coreoptes, short pedicel, funnel shape, cup shape. And Sauroptes is funnel shape, which is having a sharp edge like this. And the pedicels seems to be long, jointed pedicel. You can see the joints.
Sorry, annually funnel shape.
Here you can see the leg with unjointed pedicel means it is not having the joint, and the sucker, which is in the Sarcoptes, we indicated bell shape. Right? In Coreoptes, it is just like a cup. It is widest, and the pedicel length is very short. But in Sauroptes, it is having segmented pedicels, and the sucker, it is having sharp edges, and those sharp edges are indicative of the funnel. We are just about to finish it. Otodactylus, it's the commonest mange of the dogs and cats, and mostly they are parasitizing the ears, and that's why they are called as Otodactylus cynotus. In general conformation, resembling with the Sauroptes and Coreoptes, having ovoid body and projecting legs. So the morphology is almost similar to the previous ones with minor differences. But their preferred location is external ear pinna. Okay. Closed apodenes. What are apodenes? Any rigid integumental process that projects into the body to form the insect endoskeleton is called as an apodeme. Think about the endoskeleton. On the endoskeleton, in the anatomy, you might have observed that the bones are having some kind of points where the muscles are getting attached. Right? So in the exoskeleton, there are certain points of the exoskeleton which are embedded deep, on which some muscular things are attached. So those embedded things, these are actually rigid, integumental processes. They are projecting into the body from the outside, because the skeleton is outside. So on which you can see various structures could be attached, like the attachment of the muscles is there. Those projected stuffs are called as the apodemes. So these closed apodemes are present adjacent to the first and second pair of legs on the body. So that is a unique character in operatives, which is more pronounced. It doesn't mean that these apodemes are not present in other parts, but here they are very much pronounced and visible. Pedicels like Coreoptes are unjointed, right? Tarsal suckers, all 4 pairs of legs in male and first and second pair of legs in female. And in cats, majority of infections are acquired by suckling kittens from their carrier dams. So the transmission takes place from mother to the offspring through physical contact. It is highly contagious, and once a mother is infected, entire litters are getting infected. And the exudate from the ear canal comes out, which is brownish, waxy exudate. That is an indicator of something wrong, or the mite infestation going on in the ear. And it is usually coupled with a secondary infection which leads to intense purulent otitis. So that is inflammation of the ear, which is containing pus inside due to secondary bacterial infection at that point. Signs, what the animal will show, the sign, the frequent shaking of the head, nodding of the head, scratching of the ears with the paws, and sometimes those paws, they are also having the nails, which can lead to self-inflicted injuries, and that can open up another chapter of wounds, and then myiasis come out due to this fetid smell. Now you might be able to correlate it that from where the fetid smell will come for the primary myiasis causing flies or secondary myiasis causing flies. So these things are going simultaneously in animals. So fetid masses in the auditory canal, otorrhea, which is inflammation of the ear canal, ulceration. I hope you understand what does it mean, which is, for example, if there are more mice involved, they can lead to deep wounds inside the ear canal, inside the auditory canal, which can cause the ulceration. In dogs with normally upright ears, if the dog's having the good breeds, having upright ears, they will be going down. They externally may be true. The intensity of the itching varies. In severe cases, the external ear may be inflamed and produce pus, which is called as the purulent inflammation. And sometimes there could be perforation. And I told you that the wound that end up with the ulcer, and if there are connections between two cavities, it is caused as perforation. Now, the mites could be too much severe that tympanic membrane, which is the membrane inside the ear, जिसको हम कान का पर्दा कहते हैं, that membrane can be perforated, and that can affect the hearing capacity of the animals. So in severe conditions, torticollis, which is due to head shaking and involvement of the nervous tissue, just like in Newcastle disease, and convulsions, which is involvement of the brain, जिससे दौरे पड़ने का अमल वो शुरू हो सकता है. Excoriation of the posterior surface of the ear pinna. Ear pinna की जो बैरूनी सतह है, the outer surface, that could be sloughed off due to involvement of the mites over there, due to scratching. There could be sometimes oral hematoma. What is that? The hematoma is a collection of the blood vessel get damaged, but the outer skin is still intact. So the blood oozes out from those capillaries, but still it is trapped under the skin. So that is called as oral hematoma, between the hematoma formation on the ear canal. So ultimately, sometimes a torn eardrum is also possible in severe cases. So this is the situation of ear mange, which is the crusty and scaly ear, and that scratch wounds are also there sometimes on the lower arm. So these are the copulatory suckers, just close to the heligus in mice, and you can imagine the very long size of the bristles on the posterior side of the body. this is the situation you can see, and there are many cases which are visible in the stray dogs and cats. You can see them very well, especially in the winter seasons, because the mites they want to hibernate themselves somewhere deep into the skin tissues. The last one, which is Chilicia, which is mainly in dogs and cats, but frequent in rabbits. And there are various species. The morphology is 0.4 mm long, which is smaller, greatly enlarged palps, giving appearance to extra pair of legs. The palps, the pedipalps, which are just close to the mouth, they are so much strong that it gives an impression that there is probably an additional pair of legs. So each palp ends with a prehensile claw hair. That's again a different thing. 6 long hairs or setae on the body, 1 on each side of the terminal anus, and 2 on each side of the body between second and third pair of the legs. So this is just kind of arrangement of the 6 long hairs on the body. And legs are terminating in combs. Combs, in fleas you have talked about the combs. So similar kind of combs, they are just replacing the suckers here, instead of claws or suckers. Here you can see the claws are here on the palps. So each palp is ending up in the claw, and look at the palp, which is so much strong that it looks like an additional pair of leg. And look at these legs. These legs, they are ending up with the combs. And look at the 6 bristles. 6 bristles, which are 1, 2, 3, 4, and 5, 6. Total 6 bristles on the body of the mite. This slide is for your reading, because we have already gone through it. The sucker is present, so Sauroptes, Coreoptes, Otodectes, and different with Chilicia. You can compare them and look at that. Only this mite is showing so much robust kind of palps as compared to the others. Life cycle is similar. Mites live on hairs and fur and approach to the skin when they are requiring the feeding process. So, dermatosis in birds. Again, the roost mite or the poultry mite, the Galini, we have already talked about that. It is just 1.5 millimeter long. It is actually longest as compared to the others. It is grayish black colored, become red when engorged with blood. So this is the blood feeding mite. And it is confusing most of the cases with the tick that we have gone through, which was the soft tick, the ergasids. Soft ticks are also the ticks which are blood feeders, and they are approaching during the night times. So they are also blood feeders, and they are feeding at night. Favored habitats are poultry houses, and eggs are laid in crevices, from where the larvae and nymphs, they would be coming out of the eggs and reaching up to the host for blood feeding. What they can cause irritation, restlessness, and severe anemia. And the only palliative treatment, because you know what is the palliative treatment, which is just to increase the lifespan, because ultimately the situation may be so frustrating that if you are somehow treating the mites, the diseases which are transmitted by mites would be causing infections and damage to the birds. So palliative treatments is attention to be paid on the habitats for prevention in the future infestation. That's it. Like whatever infestation is done, it is very hard for you to control that. So why it is very difficult? As I said, palliative, because of this particular pathogen. It is Borrelia and Serena, which is causing avian spiroketosis. These are vectors. Their role as a vector of other pathogens in nature needs to be studied, but it is presumed experimentally that they are causative. They are probably vectors of Eastern, Western, and Venezuelan equine encephalitis viruses. They are viruses, vectors for the pox viruses of the fowl, and certain bacteria like Salmonella, Pasteurella, Coxiella, Borrelia, and so on. So that's why probably this is the worst scenario where they can play a role for mortality in case of birds. So this picture shows various locations, that's Sauroptic, where does it feed, Sarcoptic and Demodectic and Dermalicid, or various locations, and this also gives a kind of a size comparison from one another. History is always a good point to see to reach up to the diagnosis. Clinical signs are also helpful. Then finally the skin scrapings, and if you are having some good budget, then you can go for the blood testing and immunological tests for identification of the Borrelia inside the bloodstream. So this is a brief of the treatment. Lime sulfur dips are very good, which is 2% to 4%. The sulfur compounds are very helpful when they are mixed with paraffin. And you know the combination of paraffin for loosening of the crusty surfaces, and sulfur itself is having a caricidal efficacy at the location. Every 5 days for 4 to 6 treatments can help. Amitraz, the dipping, or any other compound which is suitable. It means the dipping can also be helpful. Injections of ivermectin, maybe the per oral sometimes is also available. Subcutaneous is also available. Intramuscular is also available. Then oral compounds of milbemycin oxime, which are also good for sarcoptic mange. And selarectin, which is a sister compound of ivermectin, is also good. And then moxidectin is also a sister with the selamectin. These are good in spot-ons. And coupled with them, never forget to give some immunoboosters so that the immunity of the host could be up to a certain level to cope up with the disease situation. So in poultry, these are the things you can go through. Spraying karni chahiye, dusting karni chahiye. There are these compounds. These are written in your textbooks. You can go through them. Then pyrethrins. Pyrethrins are another group you have already, I think, studied or read in your assignment. Then there are some, again, moxidectin and sulfur solution. They can also be helpful. All right, so I'll stop here. Thank you so much for your patience.
Thank you.